Hello everyone, this is Abebe Chukala from IHE Delft. This lecture video is on the analysis of bright spots and yield gaps. In agriculture, productivity targets refer to land and water productivity values which are attainable within productivity distributions of a crop across areas in a similar agroclimatic zone. They can be identified by applying upper percentile to the distributions of water and land productivity of a crop in a particular season and similar agroclimatic zone. Productivity targets used as a reference to identify bright spots, also to calculate productivity gaps and project production. Bright spots are crop fields that have productivity greater than or equal to the target values. The scatter plot shows crop fields based on their water productivity on the y-axis and biomass or crop yield on the x-axis. Crop fields with productivity greater than or equal to the target productivity values are colored green. The productivity beyond the target values are assumed attainable under the best technology and management, also under favorable soil and climatic conditions. Note that some of the values could also be outliers, as the productivity values are recorded due to noises and uncertainties associated with the underlying data. Thus, a close examination of the identified spots is vital to make sure that these bright spots are identified due to error in the data, or they have best management practices and favorable conditions for the crop growth. Bright spots can be traced in a project or study area. The graphs shown in these slides are examples from sugarcane producing area in Mozambique called Sinavane. The bright spots that have productivity equal to and higher than the targets are colored green. We can identify those bright spots of Sinavane scattered in a command area per a crop season as it's shown on the graph at the top right corner here. Further, the bright spot can be also traced for multiple seasons. These spots are identified as bright spots consistently in multiple seasons. Productivity gap is an indicator that gives insight on the performance of a field in a comparison with the target productivity in the area. Biomass or yield gap at individual field in an area can be estimated by subtracting the biomass or crop yield of the respective fields from the target biomass or crop yield defined in the area or region of similar agroclimatic zone. Production gap of a crop in a region of interest is therefore the sum of the yield gaps across the area in the region. WaPOR dataset can be used to assess bright spots and productivity gaps. Firstly, the water and land productivity in space and time can be estimated using WaPOR data and local information. These indices further can be used to analyze productivity target, bright spots, and productivity gaps. There are 4 main steps to run this module. Firstly, make sure the input data to the module are ready. That is, the preceding module is complete. Secondly, launch the Jupyter Notebook. The simplest option to open the Jupyter Notebook is start with opening the WaPOR WP master folder, which is downloaded from the GitHub repository. In the open folder, in its address bar, type Jupyter Notebook and press Enter key. That leads you to open Jupyter Notebook environment. Then, thirdly, open Notebooks folder, and it shows all the modules in the protocol. Finally, run the module. Here is the downloaded folder from the GitHub repository, and this one is the unzipped folder. So if we open this folder, then we can launch Jupyter Notebook here. Just typing Jupyter Notebook followed by pressing the Enter key. Okay, so all the modules are in the folder Notebooks. Here we want to run module 5. So to run module 5, so it means all the previous modules are already executed. That's the assumption, and in fact we already did. So to open module 5, just click on module 5. Module 5 has 4 major steps. It starts with setup and then calculate target productivity, followed by identifying bright spots, and then finally calculating the productivity gaps. The productivity in this case refers to both land and water productivity. And when we talk about land productivity, it can be biomass or the derivation after multiplying with the harvest index, which is crop yield. Okay, let us start with setup. So we start importing the necessary package and library, which are standard libraries and functions which are built in Python, and also some of the modules which are already prepared and placed within the folder itself. So to activate those modules, we just need to click on the cell and then select the cell and then click on the run button. So all the necessary packages are now imported. And the second step is calculating the productivity, the target productivity. So that starts with defining a function that helps to plot productivity target. So to activate that function, we need to just click on the cell that has this function and then click on the run button. The function to plot the target productivity is now activated, which will be used later just by mentioning this function name. And this is followed by calculating the target biomass and target biomass water productivity. And then for that, we need to start importing the inputs. The necessary input for this are actual evapotranspiration, the seasonal one, which are calculated under module 2. And then import also biomass, which is calculated under module 4, and biomass water productivity. To import, we just need to select anywhere in the cell and then click on the run button. So all the raster files are now imported, and then we calculate the target biomass and target biomass water productivity. So the function in this cell does the job. So to activate we click on the cell somewhere in the body of the cell, and then select the cell, and then click on the run. So now the target biomass and target water productivity values are calculated. So for each season, it's already shown here. The next one is calculating the target crop yield and target crop water productivity. So it means a target of productivity and yield, which is derived from crop yield, and crop water productivity, which is after the biomass is multiplied by, let's say, harvest index. So for this, again, we start importing the necessary inputs, which is the seasonal water consumption, the crop yield, and crop water productivity. So we select on the cell and then click on the run button. So all the rasters are now imported or activated. And then here is the stage to calculate those target crop yield and target crop water productivity. So this function does the job. So we need to select somewhere in the cell and then click on the run button. So very quick. Then we have all the values for the target yield and target water productivity, crop water productivity for all the seasons. And the third step is identifying bright spots. The bright spots are identified based on the target biomass and target biomass water productivity, and also the target crop yield and target crop water productivity. So for the biomass, first we need to import to identify the bright spot based on the biomass, target biomass and target biomass water productivity. So we import the necessary inputs. Those are actual water consumption, biomass, and biomass water productivity. So we activate the cell. That means we import the necessary inputs, and then we map the bright spots in the study area. So this the whole lengthy function does the job. So it identifies the bright spots and then show them on the map. So we select on the cell and then click on the run button. So for each season, it plots and on the graph to the left is the biomass target productivity, which is values greater than 95 percentile. The middle one is for biomass, just a bright spot or a spot that has a biomass water productivity greater than the 95 percentile. And the third column shows the graph is when the both biomass and water productivity are greater than the target values. That's why they are very smaller, because this is the third graph has a criteria that combine the first two. Now the fourth step is calculating productivity gaps. So once we have the target, we identify the bright spot. Of course, we calculated also the target productivities, the gap, the biomass. So the productivity gap is about biomass and biomass water productivity gap, as well as crop yield gaps and crop water productivity gaps. So it starts importing the necessary inputs, again biomass, and in this case again biomass water productivity, we import it, and then we create a folder to save the calculated result. So we want to calculate biomass gap. So here we gave the name 5L to biomass gaps, and for the water productivity gaps, biomass water productivity gaps, we have a name 5L2 water WPB gaps. So we can just select on the cell and then run. So it means these folders will be created under the folder data, and also in the subdirectory where the data file or the whole WaPOR master folder exists. So to calculate the biomass gap, so it means we compare the biomass at each pixel to the target, so the difference between the target and the background biomass give us the gap. So this function does calculation, and also it plotted and also save it in the folder that uh, we just created here. So we select on the cell and then click on the run button. So the calculation will be done, then will be saved in the output folder, also plotted here. By click now, we have all the graphs shown here, and the biomass water productivity gaps. Again, just comparing the target biomass water productivity and the biomass water productivity at each cell. And again, calculation will be done and then saved in the output folder, also plotted here. If we click here, then the biomass water productivity gaps will be calculated. So now the crop specific, so it means after the biomass is multiplied by harvest index, we have the crop yield, and then it means the crop yield related gap and also crop water productivity gaps will be calculated at this stage. So to do that, first we import the necessary inputs, and in this case the necessary inputs are crop yield and crop water productivity. So we import them, and then we create the output folder, where the calculated result to be saved. So in this case, we have the F5L2 yield gaps and 5L2 yield water productivity gaps, crop water productivity gaps. So we select on the cell and then click on run. So it means we have these folders now opened under the folder data. And now is the step to calculate the crop yield gap. That will be calculated just by comparing the target crop yield and the crop yield at each pixel. And then after the gap is calculated, that will be saved in the output folder. And also here, the graphs are plotted for each season. So we can select the cell and then click on the run. The job will be done. So the final then in this module is calculating the crop water productivity gaps. That is comparing the crop water productivity target to the crop water productivity at each pixel. And then after the gap is calculated, each layer saved in the output folder, which we already created, and then also plotted here. So to do that, the function help us, and then to activate it, then we click on the cell that the function is written, and then click on the run button. The crop water productivity gaps calculated, plotted, and also saved in the folder. So this is module 5 is already executed. So we can check whether all the calculated results are now in the created folder. So we have now the data. So in the data, we can check. So those folders which start with 5 are created now, and then also if we open the folder, the raster values are now calculated. Okay, so it's checked, folder created, values calculated, and also saved. And this is end of module 5. Thank you very much for listening. to this lecture video.